Hello everyone, and welcome again to microbiology. So in this chapter, chapter 6, we're going to talk about microbial growth. So as we've done in all of our other chapters, these first couple of slides are just going to be some of the information that you need to make sure that you have mastered and the textbook pages that they correspond to in your textbook. So I'm not going to read all of these things. I'm going to allow you to sort of read them, and they are definitely in your notes if you're in my class that you can download off of Blackboard. So by now, most of you all should have started to work a little bit with media, even if it's just nutrient agar, or if it's just a nutrient broth, or a slant, or a deep, not even anything that's either selective or differential. But for many of you all, you're also not only just working with media by this point in the semester, but you're also working with different types of media, whether it's selective or differential or enrichment, and understanding that different microbes would have different requirements for growth. So what this chapter is meant to discuss and explore are the basic growth requirements for microorganisms, and also to help you all realize that not all microorganisms require the same things for growth. And since we know that, those are ways for us to use biochemical tests to determine one microbe for another, since we kind of have a relative good idea of what requirements they need. So remember, when we talk about microbial growth, we're not talking about an increase in the individual organism size. We're talking about an increase in the number of microbes that are present. That's different than what we would quantify human growth, for example. For you to grow, you're actually getting taller or wider. You're getting larger physically, going from a 2-month-old infant to a 20-year-old human being. It's an increase in the actual individual. For microorganisms, we're looking at an increase in the population, or the number of microbes that are there. Microorganisms, specifically the prokaryotes, the bacteria that we work with, they grow or reproduce by binary fission. So as a result, one microorganism becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16, 16 becomes 32, 32 becomes 64, and you can see where this continues to go. So there's going to be this doubling of microorganisms in each subsequent generation. How fast that doubling takes place depends on how quickly that microorganism can go through this process of binary fission. So the results of this microbial growth are that you have a colony, which is going to be an aggregation of cells that come from a single parent cell. That's why on our agar plates, one individual colony by itself with its characteristics of being either white or cream colored or translucent or opaque, glossy, concave, whatever the characteristics were that you identified for your colony, that's why we established that all the bacteria in that colony are going to be the same species. Another type of microbial growth are biofilms, and we talked about biofilms a little bit in the first chapter, where it's not just one specific species growing in this area, there are multiple different species. It's actually a population of different bacteria, different prokaryotes usually, that are working together to create this kind of utopian environment. So it's a very complex community. So these requirements for growth can be broken apart into 2 basic categories. You have chemical requirements for growth, and then you have physical requirements for growth. Chemical requirements for growth are going to be things like nutrients, organic molecules, what kinds of carbohydrates can this organism break down and get energy from, what types of oxygen requirements are they going to need, how concentrated or non-concentrated should the oxygen be. Does it need to have a certain amount of carbon dioxide or a higher amount of carbon dioxide? What type of micronutrients or vitamins? So those are all the chemical aspect of it. The physical aspect of growth is going to be what kind of temperature can it withstand? What kind of pH can it withstand? What kind of pressure can it withstand? So microbes are able to get nutrients from a varied collection of sources. It's not like, we humans, we eukaryotic cells, where we have to get those 4 macromolecules groups. So carbohydrates, lipids, nucleic acids, and proteins. Microorganisms have a much more diverse source or pool from which they can get their nutrients. So sources of carbon, energy, and electrons for microbes, we can now, because remember we said it's a very vast area that they can get these nutrients from. So to kind of make things make a little bit more sense for us, we're going to break it down into individual categories. So we understand that living organisms are going to require some sort of energy source. We have found that as carbon-based life forms, me, you, the bacteria that are living on your tongue, that we require carbon in some form or another. That's the most efficient way to get energy. So the way that microorganisms can utilize carbon is not always the exact same. So just looking at the carbon source and how they're able to make energy from that, we have two different categories. They can be autotrophs, like plants. Remember, auto means self, so they are self-feeders, troph is feeding. So autotrophs, like plants, means that they can use photosynthesis or photosynthesis-like properties in order to make their own food. And then you have the heterotrophs. Heterotrophs are like us, that we have to eat something else. So hetero means other. So as a heterotroph, we have to eat other or get our carbon source from other things. So your apple, your banana, your bread, the chicken, whatever it is that you're going to eat. So there are some groups of bacteria that are autotrophs, make their own food. Some are heterotrophs, they have to be other feeding, eat other things. Those two groups of organisms are based on their source of energy. Chemotrophs are going to have to use chemicals in order to feed themselves. So chemotrophs and heterotrophs, they kind of go hand in hand. And then we have phototrophs that are going to use sunlight in order to make their food. That's the source of energy by which they use to make their food. So phototrophs, and photo means light, a lot of phototrophs fall under that category of autotrophs because they are self-feeding, but they have to use sunlight in order to get that energy. So here's a beautiful chart that demonstrates this. This chart is also in your textbook, where we have not only the carbon source, whether it's self-feeders, autotrophs, or heterotrophs, they have to have organic compounds. Those that are autotrophs, the carbon source that they're getting is carbon dioxide, CO2. So they're able to use that CO2 and fix it into a larger macromolecule that they can use in order to extract energy out of it. Whereas for heterotrophs, they have to use some carbon-based molecules such as glucose, lipids, other types of carbohydrates, proteins. They use those carbon sources, or what we call organic compounds. Anything that has carbon in it is considered an organic compound. They have to use those types of entities in order to get energy. So then we can look at the energy source, whether it uses light or it uses some sort of chemical. So if we look at this first box here, we have photoautotrophs. So right there in the name, we can see that it's going to use light as a source of energy, and then it's going to use carbon dioxide because it's an autotroph. So use carbon dioxide and light energy, photosynthesis should come to mind. So we have plants will obviously do that, algae will do that, cyanobacteria, those that I highlight are very important to know. They'll actually use water as an electron source to reduce carbon dioxide and produce oxygen gas as a byproduct. So we call cyanobacteria, they're oxygenic photosynthesizing microorganisms because they help to produce oxygen into the atmosphere in much the same way that plants and algae, when they undergo photosynthesis, will. We also have green sulfur bacteria and purple sulfur bacteria. Unlike the cyanobacteria, they don't use water, they use hydrogen sulfide as an electron source, and as a result of using hydrogen sulfide, they don't give us this oxygen byproduct. So they're going to be considered anoxygenic photosynthesizing organisms. But both cyanobacteria and the green sulfur bacteria and purple sulfur bacteria fit underneath this category as a photoautotroph. For chemotrophs, they're still using, these type of chemotrophs, they're still using carbon dioxide, and they're still autotrophs, so they're still self-feeders, but they don't use, they have a different mechanism by which they use it. So they can use other chemicals, they can use hydrogen, they can use sulfur, and then we have bacteria that can use those molecules and fix that CO2 or fix the carbon from those molecules to feed themselves, but they're still autotrophs. Then we have photoheterotrophs. Photoheterotrophs would be green non-sulfur bacteria and purple non-sulfur bacteria. So they have to get their carbon source from consuming other entities, but they use light energy in order to break those entities down in order to get the energy out of it. And then this is where we fit. We're chemoheterotrophs. So aerobic respiration that we talked about in Chapter 5. That is an example of a strategy a chemoheterotroph would use in order to get energy. So most animals, fungi, protozoa, many bacteria, we can have some that are not only aerobic respiration is used, but anaerobic respiration can be used for some bacteria, and then fermentation, which is used by some bacteria and some yeast, as well as some archaea. And all three of these different mechanisms by which energy can be, an organism can get energy, whether it's through aerobic respiration, anaerobic respiration, and fermentation, we discussed at length in Chapter 5. So if you're not quite sure about what these terms mean and how these processes take place, then be sure to go back and watch the Chapter 5 video, because it gives you a great discussion. And not that I'm tooting my own horn or anything, but it gives you a great discussion to help you understand those things. So growth requirements, chemical and energy requirements. So we have oxygen requirements in much the same way that microorganisms had to have some source of carbon, whether it came from carbon dioxide or it came from consuming an organic life form, such as, not necessarily life form, but an organic substance, whether it was proteins, carbs, lipids, that sort of thing. There are also oxygen requirements that have to be met. Not all microorganisms can tolerate the same amount or same level of oxygen. Some need more, some need less. So oxygen is essential, which means that it absolutely has to be present for obligate aerobes. If you're obligated to do something, you absolutely have to do it. If you're an aerobe, you have to use oxygen. So you're an obligate aerobe if you're using oxygen. For some organisms, oxygen can actually be quite toxic, because oxygen in its singlet oxygen form, unless you have the enzymes necessary to help you deal with oxygen, it can actually be toxic for you. So for us, thank goodness, we have superoxide dismutase, and we also have peroxisome. So we have an ability to deal with oxygen. So it's not toxic for us. It's actually quite useful. But for some organisms, oxygen can be deadly for them. For them, they are obligated to stay away from oxygen, and we call them obligate anaerobes. So the question that we have below is, how can this be true? Toxic forms of oxygen are highly reactive and excellent oxidizing agents. Resulting oxidation causes irreparable damage to the cells. And that's absolutely true, because it's very reactive. And if we look at oxygen on the periodic table, then what you will find is that oxygen, its outermost shell of electrons, is missing a couple of them in order for it to be stable. So as a result, because it's missing those two electrons in the outermost shell, it's very, very reactive. It's not stable. It's going to want to pull two more electrons, because that's how much it's missing. It's going to want to pull two more electrons in its outermost shell in order to be stable and reactive. It's very easy to borrow or share those two electrons from two hydrogen molecules, and that's how we get H2O, or water. So in order to deal with oxygen in its normal form, that single form of it, the single molecular form of oxygen, then organisms have to have certain enzymes to help deal with those free radicals that can be made and that toxic form of it. And those two enzymes that are required, two of the three enzymes that are required, are superoxide dismutase and then peroxidase. So the four forms of oxygen, the superoxide oxygen, the superoxide radicals, the peroxide ion, and the hydroxy radical, these are all those toxic forms of oxygen. So if a bacteria doesn't have a strategy, as I said before, in place to deal with these toxic forms of oxygen, then oxygen is going to be toxic for it. It's not toxic for us, because we have enzymes that are required to deal with these toxic forms of oxygen. Some bacteria, like us, those that are obligate aerobes or those that are facultative aerobes, that means are facultative anaerobes, they have some, if not all, of the enzymes that are required to deal with oxygen in its toxic form. So many organisms can live in various oxygen concentrations. We have aerobes. Obviously, they can use oxygen, it's kind of in their name. Anaerobes, some that can deal with certain amounts of oxygen, not too much of it, but they prefer to get their energy in areas where there's no oxygen that's present. Facultative anaerobes, they can go between both of them. They can use oxygen if it's present. It's a more efficient means of getting energy if oxygen is present. If oxygen is not present, then they can switch over to fermentation and get energy that way. Aerotolerant anaerobes, which means that they will Aerotolerant aerobes, which means that they can deal with certain amounts of oxygen, so it means they have some of those enzymes that are required to deal with oxygen in its toxic form, but not all of them. And then microaerophiles, they only need a little bit of it. So what we're looking at here are tubes that you will probably use in lab class. You'll definitely use it if you're in my class. And these are called thioglycolate growth medium tubes for oxygen. So what we're looking at here is that in this tube, there's a high amount of oxygen concentration at the top of the tube, and then it progressively decreases as we get to the bottom of it. So what we can do is if we don't know the oxygen requirement of the microorganism that we're working with, then we can put it in one of these thioglycolate tubes and observe the area of growth. If there's a high concentration or a high amount of growth that's happening at the top of the tube, then we would consider these obligate aerobes, because they have to be in an area where there is lots of oxygen that's present. We call this type of media a reducing media because the oxygen concentration is going to reduce as we go from the top of the media down to the bottom of it. For obligate anaerobes, they can't deal with oxygen. They don't have any of the enzymes available to deal with oxygen in its toxic form. So as a result, we should expect to find them growing at the bottom of the tube, where there is the lowest or the least, if any, oxygen that's available. Facultative anaerobes, notice how they're growing throughout the tube. Now for facultative anaerobes, even though they're growing throughout the tube, there's a higher concentration of them at the top of the tube. And the reason for that is because if you have some, even if you don't have all of the enzymes to help you deal with oxygen in its toxic form, oxygen is by far the most efficient means to get energy. If you remember back from our discussion in cellular respiration in Chapter 5, that if you used oxygen or used a process of cellular respiration or an oxygen-containing compound like a nitrate, sulfate, carbonate, then you got 34 ATP from just one step, just from the ETC, the electron transport chain. So if you can use oxygen, it's much more beneficial because you're going to get much more energy as a result. However, if the you-know-what hits the fan, then you can have an instance where if there's not enough oxygen present, these facultative anaerobes, they can switch over to fermentation or switch over to strategies that don't require the use of oxygen or oxygen-containing compounds. For aerotolerant aerobes, notice that they're going to be evenly distributed throughout the tube because they can tolerate oxygen. They don't really have a preference for no oxygen or oxygen present at all. So the next nutrient is nitrogen. So far we talked about oxygen and carbon nutrients, and then the energy source. So the energy source can come from sunlight. The energy source can come from organic molecules. We talked about oxygen, different oxygen requirements. Now we're going to talk about nutrient requirements. So anabolism, if you'll remember, that's the building up of things, often stops as a result of not enough nitrogen that's present. Nitrogen can be acquired from organic or inorganic nutrients. And all cells are going to recycle nitrogen because they need it as part of the amino group to make up their amino acids, and they also need nitrogen to make up their nucleotides. Amino acids build proteins. Proteins are vitally important for all bacteria. All living things, in fact, they require proteins because those proteins can act as enzymes to help with metabolism. They can be part of the cell wall, that peptidoglycan layer. We need them for that. So proteins are vitally important. So being able to use nitrogen and having nitrogen available is an absolute requirement. And how much of it does bacteria A need from bacteria C or D? That can vary because they can be two different bacteria, but they do require some amount of it in order to make proteins and also to make copies of their genetic material, DNA and RNA, so that they can have the populations grow. Nitrogen fixation can happen by certain bacteria, and it's essential for all life here on Earth. So not only do the bacteria benefit, but we benefit from it as well. And what nitrogen fixation is, it's just pulling the nitrogen out of the atmosphere and then fixing it into various different compounds. So other chemicals that are required, we need phosphorus. Phosphorus typically for proteins as well as for nucleotides, and we need it definitely for ATP. We need some inorganic phosphate floating around for that phosphorylation to take place. Sulfur helps to build those disulfide bonds between proteins. And then for trace elements are those molecules that are only needed in small amounts. And even though you only need a little bit of them, there are still requirements for growth, because if you don't have those trace elements, then the bacteria is going to not function optimally, and it's going to compromise the growth in the colonies. Other growth factors are things like coenzyme 10, which can be used in the electron transport chain. They're necessary organic chemicals that the organism must get from the environment, and they can't be synthesized themselves. And which growth factors can't be synthesized by all bacteria? Well, it just depends on what bacteria we're talking about. Some bacteria can synthesize certain growth factors, whereas others cannot. And by having that knowledge, that's another key piece of how we can run biochemical tests to identify unknown bacteria. So on to physical requirements. So we talked about some chemical requirements and the need for nitrogen, growth factors, trace elements, oxygen, as well as carbon dioxide. Now are some physical requirements. So physical requirements, first one that we're going to talk about is temperature. Temperature can affect the three-dimensional shape of those proteins. And remember, we talked about that protein form dictates protein function. And if a protein loses its form, it's also going to lose its active site, so it won't be able to function properly. So the substrate won't fit onto that active site because it's not there anymore, and then that substrate won't be broken down or built up by the protein or the enzyme, the specific type of protein. So temperatures that are too high can cause the protein to become what we call denatured. So let's highlight that. Temperature causes denaturation. That's what that basically says. Lipid-containing membranes of cells and organelles are also lipid sensitive. So if the temperature is too low, and you guys know this, if you take oil and you set it outside or set it in a refrigerator, it's going to cause it to become much more rigid and more stiff, and then subsequently it can also become more fragile. These plasma membranes or anything that contains lipids, if it's too high, then it's going to become too fluid, so it's going to move too much. So not only does temperature affect proteins, but it can also affect lipid-containing components of the cell. So here we're looking at a chart that's showing the effects of growth and microbial growth, the effect of temperature on microbial growth. So where we see the highest point, the highest zenith, and we look at the y-axis and we know, we recognize we're going from 0 to maybe, I don't know, 10, cells per hour or whatever it is. But it's just a generic graph that we have as the growth rate increases on this side, the highest point of it, the highest amount of growth rate is going to happen at a particular temperature here. And we consider that the optimal growth temperature. Whereas you may have a little bit of growth that's happening down here at this lower temperature, and maybe this lower temperature is 22 degrees Celsius. So we'll have a little bit of growth that's happening there. And the same thing on the maximum growth. We're really not getting any growth at all at this point. The bacteria probably are dying because it's way too warm. But even here, at a little bit higher growth or higher temperature, you're just not getting nearly as much growth because the temperature is just too high. So the optimal growth temperature, and especially for those bacteria that cause disease or are pathogenic, is usually about 37 degrees Celsius, where we'll notice on our plates, we incubate this plate at 22 degrees Celsius, we have some growth, but not a lot. At 30 degrees Celsius, we have a little bit more, but at 37 degrees Celsius, we have much more growth that's taking place because this is the optimal growth temperature. Now, just the same way that we said that not all bacteria require the same amounts of oxygen, not all bacteria have the same temperature that's going to be their optimal growth temperature. So 4 temperature ranges we have here, and I'm going to highlight these because I believe they're important. So we have psychrophiles, which like low temperatures, typically refrigeration temperatures, so bacteria that grow in the refrigerator. So you've got that, Tupperware dish of peas that you haven't touched in a few weeks, and I'm sure I'm not the only one that this has happened to. They're stuck in the back of the refrigerator and you see them, they're starting to smell a little weird. Those microorganisms that are growing on them, we would consider them psychrophiles because they can handle those cooler temperatures. Mesophiles, they can handle temperatures, they prefer temperatures right here at about 37 degrees Celsius, which is human body temperature. So lots of microorganisms that cause disease or are pathogenic are mesophiles. Thermophiles are going to be those microorganisms that like much warmer temperatures, so about 65 degrees Celsius here. And keep in mind that water boils at 100 degrees Celsius, so they can handle much, much warmer temperatures. And then we have those thermophiles and hyperthermophiles are typically archaea. So with the hyperthermophiles, these are the guys that can handle really, really hot temperatures. So archaea, many of the organisms in the domain archaea are what we call extremophiles, and they are either thermophiles or hyperthermophiles, and they like really, really, really warm temperatures. Not only do they like it, but they can actually tolerate those really warm temperatures. Fortunately for us, because we use temperature as a way of controlling microbial growth, that's why we incinerate or flame our loops when we are sterilizing our loops to make when we work with our cultures in lab. Fortunately, most archaea aren't pathogenic. pH is another physical requirement. Much the same way that temperature can cause a protein to lose its three-dimensional shape and denature it, pH can also do the same thing. Hydrogen ions can interfere with hydrogen binding, and that also is another way that it kind of screws with the ability of the microorganisms to undergo metabolism. If a microbe can no longer do metabolism, break stuff down, and then build that stuff back up to be in a useful form from the bacteria, bacteria dies. So Neutrophiles are going to grow best at a neutral pH, and remember the pH range goes from 1 to 14, where 7 is considered neutral, so it's right there in the middle. 7 is neutral, and I'm going to change my pen color just a scotch. And anything above 7 is considered alkaline or a base. And things that like pHs that are above 7 are, they live in alkaline soils and alkaline water, so anything that's got a pH higher than 7. And then acidophiles, change its color, grow best in acidic habitats, and anything that's an acid has a pH lower than 6, and we call those acids. So there's that whole acid and base thing you've probably learned in an introductory biology class or in chemistry. Water is another, physical requirement of microorganisms. Microbes require water to dissolve enzymes and to dissolve nutrients for hydrolysis to take place. Lysis breaking apart hydro, water, using it to it. Water is absolutely essential for lots of metabolic properties. Most cells actually die if they become desiccated. Without water, in the absence of water, they die. Some cell walls retain water as a way to deal with moments of drought when there's not enough water present in the environment. Some will actually form endospores, which we talked about, or cysts. And those endospores and those cysts, they cease all metabolic activity and they just kind of lie dormant until the conditions get better. So the two physical aspects of water that we're going to talk about and how they relate to microbes, microbial growth, are both osmotic pressure and hydrostatic pressure. Now, osmotic pressure you're probably very familiar with because we talked about how the cell wall of gram-positives and gram-negatives is meant to deal with osmotic pressure by not letting too much water get out and not letting too much water get in. So osmotic pressure is the pressure that's exerted on the semi-permeable membrane across a solution where solids cannot freely cross the membrane. So that's what we define osmotic pressure as. Hypotonic solutions have a lower solute concentration. As a matter of fact, let's highlight that. So if we place a cell in a hypotonic solution, remember there's not a lot of solids in there, so not a lot of sugar, not a lot of salt, not a lot of little particles. Those dissolved particles are the solutes. And the solvent is the water, so lots of solvent, less solute. So in fact, let's write that down. Let's say 99% water here in a hypotonic solution. So a cell that's placed in a hypotonic solution is going to swell. That means water is going to go into it because the cell, let's say it only has 90% water. on the inside. And remember, water goes from very high concentration to low, so from the solution that the cell is put into to inside the cell. Hypertonic solutions are different. Hypertonic solutions, let's say that they have, and I'm being drastic here, 80% water, and then let's keep the cell at 90% water, and then 10% solutes, which is, like I said, this is drastic, but I'm just doing this to illustrate a point. So we still have, now at this point, we have a higher concentration of water inside the cell than we have outside the cell. There are more solutes in that hypertonic solution, more salt. So if you were to taste it, it's going to be saltier. Or it's going to be really, really sweet, like the Kool-Aid that I made. It's, you know, very little water and lots and lots of sugar. So the concentration of the water is higher in the cell than it is in the surrounding solution. So the water is going to leave out of the cell, going from an area of high concentration, at 90%, to that area of low concentration. So the cell is going to shrivel. Some organisms, some microbes, actually can live and prefer an environment where it's very salty. And what we call those organisms is that we call them halophiles. Some organisms actually require a very salty or a hypertonic solution to live in, so we call them obligate halophiles, whereas others can just kind of tolerate it. If it's like that, they can deal with it. If it's not, they can also deal with it as well, and we call those facultative. So hydrostatic pressure, what we just talked about was osmotic pressure. So hydrostatic pressure is the pressure that the water exerts pressure in proportion to how deep it is. So this is going to be looking more with pressure and water. So looking at the combination between the physical aspect of pressure and water. Those that are considered barophiles, which means they can live in very extreme pressures, those microorganisms that can live at very, very deep canyons and ravines of the ocean, those would be considered barophiles. Their membranes and enzymes actually require this nice, steady, much higher than atmospheric pressure in order to keep the protein's three-dimensional shape. So if you don't have that amount of pressure, then those organisms will die. So organisms can live in association with other species and form biofilms, and what we're going to talk about have three different relationships. If they are forming biofilms with them, they're usually living in a synergistic relationship, which means that I scratch your back, you scratch mine sort of thing, where my trash is your treasure. We kind of work together in that sense, and that's one of the reasons they have what's called quorum sensing that happens with bacteria, that they will actually communicate with one another through chemicals. A lot of times, organisms can have an antagonistic relationship with one another, which means kind of like they're warring. They're either fighting over resources, they're fighting over space, they're fighting over nutrients, but one is going to outcompete the other. And a symbiotic relationship, remember we talked about the endosymbiotic theory between what we believed mitochondria and mitochondria and chloroplasts were their own prokaryotic cells, and then larger cells engulfed them, larger eukaryotic cells engulfed them. And they formed a symbiotic relationship where the larger cell provided protection to the smaller cells, and then those smaller cells, chloroplasts and mitochondria, or pre-chloroplasts, pre-mitochondria, they provided energy toward it. So they worked well with one another. They weren't fighting with one another. So biofilms, as we said before, are just very complex relationships among numerous different types of bacteria. They don't necessarily have to be the same species. We find biofilms in lots of different places. In the ring around your toilet, there's a biofilm there. In your shower, there's probably a biofilm there. On medical devices, on mucous membranes in the digestive system. So there are lots of places that you can find them. Biofilms on medical devices such as catheters can be problematic because it can cause a nosocomial infection or hospital-acquired infection, and they're sometimes difficult to treat because there's more than one type of bacteria there. So you have to use a very broad spectrum antibiotic to treat those types of infections. As I said before, bacteria that are in, or microorganisms that are in a biofilm, they communicate with one another, and they communicate with one another through this quorum sensing. Many microorganisms are more harmful as part of the biofilm, and scientists are always looking at ways to prevent biofilm formation, especially on implanted medical devices. So now on to the culturing aspect. Now that we've talked about the physical requirements for growth and the chemical requirements for growth, let's talk about how we can simulate these environments in the laboratory. So a little bit of terminology. Inoculum is introduced into medium. Inoculum is going to come from your culture. That's the bacteria, those prokaryotes that you want to put on this thing that you want it to grow on. So that's what we call our medium. We can have clinical specimens or stored specimens, or you can get specimens from the environment. So you can just go swab a toilet seat or swab the floor, and that would be an environmental specimen. Clinical specimens would be specimens that come from your patient. And stored specimens are what we work with in lab, and that, you know, we just have E. coli on deck. We've got salmonella on deck. We've got these different organisms that have just been stored. A culture, or to make a culture, is the act of cultivating microorganisms or the microorganisms that have been cultivated. So when we go into lab, our cultures are already done by our lab coordinator, and it's probably the same at your college or university, that your lab coordinator has already made the culture, and you would use that culture to inoculate your media. So notice how I'm using this terminology. Your culture will inoculate your media, and your inoculum is the culture that you place on your medium. So that inoculum is introduced into the medium. The inoculum is made up of your culture, which are your bacteria. Hope that makes sense. So we know, and you've probably all seen this, that not all colonies are the same. They have different shapes. They can be irregular, they can be filamentous, they can be spindle, they can be flat, they can be convex, they can be craterform, they can be lobate for what their edges or margins look like, they can be small, large, smooth or rough, shiny or dull, pigmented or non-pigmented. So there's a whole lot of characteristic terms that we can use to describe how these colonies are growing. And many times the shape of the colony is going to be consistent for every bacteria in that species. So in order to obtain a pure culture, which is a very vital part of your unknowns, there are certain things that we want to do. Pure cultures are going to be cultures that consist of just a single cell that's given a lineage to all these other cells. So everything on this plate is the exact same thing. So a progenitor is a term, is termed a colony-forming unit. So the CFU, or the colony-forming unit, is one particular species of cell. So everybody else in that colony is going to be the same thing. In order to maintain this pure culture, we want to use aseptic technique. So that is the flaming of your leaf, letting it cool, and then transferring only that particular colony, nothing else, onto a fresh agar plate, so that only that particular bacteria is going to grow. So there are two common isolation techniques that we will use, or we'll talk about, but there's only one that we really use, and that's streak plates and pour plates. Streak plates, I'm going to highlight that one because that's the one that we routinely use in lab. So the pour plates here, we're just kind of looking at basically a serial dilution, and notice how we went from the initial sample or the initial culture, we put it into 9 mils of broth, 1 mil from there to the next, from there to the next, and we noticed that the higher our dilution rate is, the lower, the fewer the number of colonies that we'll find growing on the plate. And that makes it very easy. Notice how you have all these different types of colonies, and it's going to be difficult to, if you wanted to get this guy here, it might be sort of difficult if he may be contaminated from this colony or that colony touching it. This colony would probably be easier to transfer because it's sitting by itself. So you want to pick colonies when you're doing your isolation. You want to pick colonies that don't have other bacterial colonies growing very close by, so that you can lift it from that plate and put it on its own plate, so that on the next plate that you make, there'll only be that bacteria that's growing there. So it's going to be much easier to pull a colony from this plate aseptically than it will be for this. So serial dilutions can help with that. Some fungi isolated with a streak and perchlade, so we can use that. Protozoan and motile unicellular algae are isolated through dilution of broth cultures. And at that point, as I said before, it's all kind of repeating. Then you can individually pick up a single cell and establish a culture from that. And you can use that as your stored culture. Culture media, most prokaryotes, in fact, all that we're going to work with have demonstrated it can be grown on culture media. A majority of prokaryotes have not been grown in culture media, but those that we call E. coli are microbiology lab pets because we always work with it. We can use a variety of different liquids or solids to culture microbes. The most common kind are nutrient agar and nutrient broth. We use agar in our Petri plates because it's a nice media to work with, because most of the microorganisms that we work with can't break down that agar. They can't break the polysaccharide, so they can just grow on it nicely. So the different types of general culture media that we are going to talk about are listed. So we have defined culture media, complex culture media, the type that we work with in lab. That we work with in lab recently and will continue to work with are selective and differential media, anaerobic media, and transport media. So all of these different media types we will be discussing. So for what we're looking at here, this is a synthetic defined media. So this is, we call it defined media because we can quantify each one of the chemicals that are going in here, so into the media itself. So it's very, very specific on exactly what we're looking for. It's all, everything has been chemically defined. Now you're probably wondering, well, why didn't they chemically define glucose? Well, glucose C6H12O6. That's kind of a standard. It's kind of like water. Everyone knows the chemical formula for water, H2O, so there's no reason to write H2O out in its molecular, its chemical form. Same thing with glucose. If you're a scientist and working in microbiology lab, you should know what glucose's chemical formula is. So that's why they haven't put that there. So difference between defined media and complex media is that defined media is going to literally chemically spell out what each one of the components that make up the media are. It's not going to just say proteins or beef extract or calcium carbonate. It's going to actually spell it out and also give you the exact concentration at which you can find the media in there. Now with complex media, the exact chemical composition is unknown, so we're going to highlight that. Whereas with the defined media, we knew the exact chemical composition of it. So complex media is what we would consider we work with in labs, that we don't know all of the exact chemical composition of the components of the media, but we do know that there's more than one thing in there. Complex media has just got more than one thing that makes up the media. So you might have some glucose, beef extract, and throw some amino acids in there. What amino acids? We're not specific. We're just saying some amino acids are on there. Complex media is really helpful to grow a variety of different microorganisms. And we can use complex media when we don't know exactly what the nutritional requirements of the bacteria are. So here we have just blood agar. Notice that it says agar we have in here. We have pancreatic digestive casein. We've got some water. We've got some sterile blood. We've got some salt. For McConkey's agar on here, we got some peptone, we got some agar, some lactose, some biosalts. Notice that we just said biosalts. We didn't put the molecular chemical composition of those biosalts. So both of these could be considered a type of complex media. Both your McConkey's agar and your blood agar, not only are they complex, but they're also differential. That means that different types of bacteria that are grown on this media are going to cause the media to look differently, or the bacteria are going to appear different on this type of media. So that's why we call it differential. And it's complex because it's got more than one thing in it. So to culture anaerobic organisms, we would use what's considered an anaerobic culture system jar or anaerobic jar, a gas. We'll make a gas chamber here. What we've done is that we use this to grow organisms that maybe don't tolerate oxygen, that they need to be in an oxygen-free system. So we put in there an envelope that contains chemicals that will release carbon dioxide and hydrogen, rack our Petri plates upside down, and then this methylene blue is our indicator for whether or not we have achieved this aerobic conditions on here. So when we have this hydrogen and this carbon dioxide gas that comes out, then it will be attracted to the pellets that are in here, and it removes all of the oxygen from this chamber. And it causes it to be anaerobic. To preserve cultures, we can refrigerate them, deep freeze them, or we can use lyophilization, which is freeze dry them. Lyophilization allows the cultures to be used for decades. You can store them for a long time. Deep freezing them allows us to use them for several years. And refrigeration is useful for just short periods of time, like 6 weeks at most. When we're ready to preserve our cultures, we use refrigeration in the classroom, and our lab coordinator will deep freeze or use lyophilization to store cultures for longer. That's of economic significance for us because it allows us to continue our culture lines without having to constantly buy new cultures if we can just use our same cultures from year to year or semester to semester. So another aspect of growth, which we kind of touched on at the very beginning of this discussion, was how microorganisms are, they grow using binary fission. So I'm going to let you all look at this for just 1 second, and then we'll talk about it. So it's pretty simple, right? One microorganism is going to become 2, and that's basically what we're having happen here in binary fission. That you have this microorganism that will double its chromosome, this chromosome singular, and then after it doubles its chromosome, you're going to have the cell wall and the cell membrane invaginate or split between the 2, and this one cell becomes 2. And in a nutshell, that's pretty much what binary fission is. You do not reproduce via binary fission. Binary fission is a form of asexual reproduction, not of sex and not using sex, and you are a sexually reproducing organism. So you're going to have to require someone else's genes in order to make another member of the next generation. So since we know that bacteria are going to divide, one cell becomes 2, we can take that information and we can do some really cool things with it. Like we can determine what is the generation time. And understanding what the generation time is important to help us to learn how to control microbial growth. So the generation time is defined as the time that's needed for a bacterial cell to grow and divide and put another cell into the subsequent generation. Now, this generation time can be dependent on different factors. As I said before, if we know the generation time, then we can figure out ways to control microbial growth. So it's dependent on both chemical and physical conditions and on the species of bacteria. Something like Staphylococcus aureus has a generation time of like 20 minutes, whereas Mycobacterium tuberculosis has a generation time of like 18 hours. Like it takes 18 or 20 hours to get a new generation of Mycobacterium, where it only takes 20 minutes to get a new generation of Staphylococcus aureus. So not only does it depend on chemical and physical conditions that these bacteria are growing in, obviously in optimal conditions, you're going to have the optimal, it's going to have a generation time as fast as it possibly can, but it also depends on the individual bacteria themselves. So what we're looking at here are the different ways that we can categorize or we can graph the amount of cells that have grown in a culture in a set amount of time. So the first way that we're looking at it is just by using the raw data and the raw numbers. The second way that we're looking at it over here is that we've actually putting into its logarithmic scale. So we're looking at the log of this. Now, for this one, the story is still the same. So we're still seeing overall that there's an increase in the number of cells over time, right? So from 1 hour to 5 hours is an increase in cells. From 1 hour to 5 hours is an increase in cells. What this one's showing you as the raw data is that we're actually looking at the exact number of cells. So we're showing this doubling where 1 becomes 2, 2 becomes 4, 4 becomes 8, 16, 64, 5, 12, all the way up to what we're individually counting. So what happens here is that we're individually counting all of these cells. These kinds of numbers can be quite cumbersome, and it can be a very huge burden to actually count all these individual cells and to quantify all these individual cells. When this graph, when we use the logarithmic version of it, this is much more easier, this information is much easier to digest, and then we have just like a nice cleaner graph that we have here. So we prefer to use the number of cells in the log scale as opposed to using the actual number of cells. So that's what we're going to use, the log base of the cells, as we calculate the number of cells in a generation and calculating generation time. So for those bacteria that are growing this, you know, whether you calculate it or quantify it by the actual cells or through the log of them, they're going to have 4 basic aspects of their growth curve. They have the lag phase, and at the lag phase, the number of cells, so they're still dividing, they're still growing, but it's slower. 1 becomes 2, 2 becomes 4, 4 becomes 8, and so forth. So you're not seeing this huge, robust increase in cell numbers right away. So in the lag phase of it, the cells are kind of just preparing for the division to take place. The nucleic acids are doubling, are making copies of themselves. They're having the cell have the invagination between the cell wall and the plasma membrane to make those 2 cells. So we call that the lag phase. In our exponential phase, or our log phase of it, this is where we're going to have the most growth that's happening there. So we're going to have a huge jump in cells when we have this exponential phase take place. And then at our stationary phase, well, this is where the number of cells entering the population is roughly equal to the number of cells that are exiting out of the population. So at this point here, the number of cells that are present is going to basically stay the same. We're not seeing any growth and we're not seeing any death yet. And then at some point, we're going to have a decline or a death phase, where the nutrients, the pH, the toxins that are available there, because you have all of these microorganisms that have their byproducts, that's going to not be conducive to the growth of the microorganism. So at the death phase, we have more cells that are dying than we have that are coming into the population. And it's definitely not staying stationary. So ways that we can measure this microbial growth, we can use microscopic counts of it. That's a direct method, and it doesn't necessarily require any incubation. To do that direct count, we can use electronic counters, or we can, in those electronic counters, we can use a Coulter counter, which counts cells as they interrupt an electrical current. It's going to kind of be a little ding that it hits there, so every time a cell crosses that current, we can count them that way. We can do flow cytometry, which detects the changes in light transmissions as the cells pass a light detector. So kind of like a little blind spot hit, so they count that as 1. And then you have the media that's flowing through here, so you make sure you only count 1 cell twice. So these are the electronic versions of counting. You may have a cell, colony counter in your lab, which is like a microscope, and it's got a grid on the back of it, and you set your plate there and you can count the colonies and you can estimate about how many cells are there, but that's very tedious. Most of the ways that if we're going to do a direct microbial count, there'll be some sort of electronic counter that's going to be used, just much more efficient. This is a direct method that requires incubation. We can use a serial dilution and do plate counts from that. We can use membrane filtration, and it once again requires incubation, so the microbes have to actually grow from a time. And we can also use most probable number to give quantitative data on about how many cells we have there. So here's that serial dilution method that we have done in class. Remember, we take our original inoculum, put 1 ml of it into a broth containing only 9 ml, and then we'll take 1 ml of that into another, so that we reduce the concentration of the microbes by 10 every time. So the first time is 1 out of 10 ml, the second time is 1 out of 100 ml, and then 1 out of 1,000 ml, and so forth. We can take those numbers and we can put them use the exponential form of those numbers. And when we use the exponential form of those numbers, we can count the number of colonies that we find on the plate and then multiply it by a serial dilution number, and that's going to give us a rough estimate of about how many bacteria are present there. And this is another way we can use quantitative quantitative data to determine about how many bacteria are there. None of these methods are going to be 100% accurate except for the electronic counter, and even then there's still, you still have a small margin of error. So indirect methods of looking at microbial growth, this is more qualitative. It's going to be looking at turbidity. How cloudy is it? We can look with the naked eye and say, oh, this is pretty cloudy. It's much cloudier than this test tube, so there's more microbial growth that's happening in this test tube as opposed to the other. But a more scientific way to do that is use a spectrophotometer, which will measure the exact percentage of light that can pass through the test tube, or it can actually measure how cloudy it is by looking at the amount of light that passes through the test tube, or you can measure it by how much light was absorbed onto the light-sensitive monitor. So this test tube is obviously not nearly as turbid or cloudy as this one is. So if we put this into the spectrophotometer, then you're going to have 100% light transmitted. Whereas if we put this test tube through there, not all of it's going to be transmitted because some of that light's going to actually bounce off of the bacteria that are in there, off of the stuff that's making it cloudy, and it won't all be transmitted to the light-sensitive detector. So as a result, we would say that the percent of light that's transmitted is much lower, or there's a much higher rate of absorption of light by the bacteria that are in there. And you use that as your quantitative or qualitative data to determine that, yes, there's microbial growth that's happening in here, and this microbial growth is higher because there was much more light absorbed by the presence of the high number of microbes that are in there, as opposed to this one where it had a much greater percentage of light that was transmitted because there was nothing in it. Other indirect methods of measuring microbial growth aside from turbidity are looking at metabolic activity, measuring how much of the acids are there, and that's usually done, as I said before, it's more of a qualitative type of measurement. The dry weight of the microbes after you freeze them and you use lyophilization, or you can isolate DNA sequences of using genetic methods as another way of calculating microbial growth. All right, so that is it for the session, and I will be seeing you soon. And in the next session that we go over, we are going to talk about microbial genetics. I know, fun stuff in Chapter 7. All right, well, have a great morning, noon, or night.